Hello, my name is Yanbin Yin from University of Nebraska. Today, I want to introduce AcrFinder and AcrDB, two bioinformatics tools that we have recently developed for anti-CRISPR research. So both tools are available online at two websites. The address is shown here. I want to thank three PhD students who are the persons behind the work I'm presenting today. I also want to thank NSF and NIH for supporting our research. A quick outline, I will be introducing anti-CRISPR, their sequence features, and then talk about bioinformatics approaches to the discovery of new anti-CRISPRs with three ideas. So what are anti-CRISPRs? So the first anti-CRISPR protein was discovered by Bondi Dinami and Davison in 2013 in some phage genomes which have anti-CRISPR-Cas functions. As shown in the left figure, Cas enzyme of bacteria can make the cut in the phage DNA guided by CRISPR RNA. Shown in the bottom is the diagram of CRISPR-Cas loci, which contains Cas genes and CRISPR arrays, shown as small diamonds and squares. However, some phage genome contains AcR genes. So those AcR proteins shown in the middle of a figure as red triangle binds to Cas to inhibit cutting phage genome, so that phage will survive. So interestingly, many AcR genes have a downstream AcR associated or ACA genes to form an operon. In 2019, a Cell paper found that ACA actually is a transcription factor that represses the expression of AcRs. So as we all know, CRISPR-Cas technology is being widely used in mammalian and plant genome editing. However, many people forget that CRISPR-Cas itself is only found in bacteria and archaea, but not in eukaryotes. As the natural inhibitor of CRISPR-Cas, the discovery of anti-CRISPR has immediately caught the attention from the genome editing community. So this Nature method review paper by Bondi Dinami last year summarized many great applications of anti-CRISPRs in genome editing. As mentioned, CRISPR-Cas loci of bacteria encode Cas enzyme, which cut the phage DNA. So Cas with CRISPR RNA form a protein RNA complex, and the CRISPR RNA will guide the Cas enzyme to its right target site. On the right, we see that some phage can encode anti-CRISPR genes. Those genes encode short proteins, usually less than 150 amino acids, shown as those small boxes, circles, triangles in different colors. So this AcR protein inhibit Cas binding or cutting DNA by binding to Cas protein or their DNA target. These are some of the known mechanisms, but more novel mechanisms are being discovered. Since 2013, 89 AcR proteins has been experimentally characterized to inhibit 11 CRISPR-Cas subtypes. So when I see 89 proteins, I actually mean 89 protein families classified based on sequence similarity, so that between two different families, there's no sequence similarity. When taking these 89 proteins for sequence homology search, most of them have homologs restricted to genome of the same species. And most of those proteins do not have a PFAM domain. Out of the 89 AcR proteins, 16 have their three-dimensional structure solved. Interestingly, they all have different structural folds. So this means that they all have a different evolutionary origin. So interestingly, although without sequence and the structural similarity, they all come to the same function, which is inhibit CRISPR-Cas. In 2019, we look at the sequence features of experimentally characterized AcR proteins. We can see here 80% of the AcR proteins are shorter than 150 amino acids. 60% of them have ACA gene in the gene neighborhood to form an AcR-ACA operon with very short intergenic distance. So even the ACA protein are also very short. Most of ACR proteins are negatively charged. So here's a graphical representation of the operons that encode some of the 89 known ACR proteins. As we see here, most of the ACR proteins form gene operons, with ACA shown in light green, also with other ACR proteins shown in other colors. If they do appear in multiple genomes, for example, the ACRIC6 in dark green, they tend to be restricted to genome of the same genus, in this case, Pseudomonas bacteria. They also tend to be found in prophages, and therefore there are phage-related genes in the gene neighborhood. So to summarize, there are some key sequence features of ACRs and their operons. They are very short, tend to form multi-ACR gene operons with more than one ACR genes and sometimes an ACA gene. So the ACA protein actually is more conserved. They contain a conserved HGH PFAM domain, as they are often found in phages and the prophages. Their gene neighborhood tend to have phage genes and functional unknown genes. So we'll come back to this table later. So in order to identify new anti-CRISPRs, people have been using two bioinformatics approach. The first one is called GUIT by association. That is, identify the more conserved ACA genes first by homology search, and then experimentally characterize the upstream genes to see if they have anti-CRISPR-Cas function. So many of the known ACR genes were found by using this approach with iterative search of ACR and ACE homologs. The second approach is called self-targeting spacer, or STS. So the idea is that, shown on the right, if a bacterial genome contains CRISPR-Cas and its CRISPR spacer has some identical matches elsewhere in the genome, so that means the CRISPR RNA can guide Cas enzyme to target its own genome, so which will lead to autoimmunity and kill the bacteria itself. So to avoid cell death, the cell must have a mechanism to inhibit its own CRISPR-Cas. Therefore, looking for anti-CRISPRs in bacteria with self-targeting spacers will more likely to succeed. So we have combined the two approaches in one bioinformatics pipeline shown in this diagram. So given a bacterial genome, we will look for ACE homologs and then locate them in small gene operons, which has enrichment of mobile genetic element genes in the gene neighborhood, also tend to be found in genomes with self-targeting spacers. So we have further developed AcrFinder, a computer program that automates this Anti-Crispr discovery pipeline. So using 8 published ACR-ACE operons as the testing data set, we have shown that AcrFinder has a good recall in leave one out experiments. So as of today, there are 9 online bioinformatics tools for ACR research. In addition to AcrFinder, there are 2 other programs, ACR Ranker and AcrCrispr, also allow to perform automated ACR discovery given protein sequences or genome sequence. However, AcrFinder is the only one focused on ACR-ACE operon identification. So 3 tools use machine learning, ACR catalog, ACR Ranker, and AcrCrispr. So come back to this table. The 4 ACR discovery tools all use these sequence features. Among them, ACR Ranker is the only one that uses biased ACR amino acids chimeras. ACR catalog combines multiple features to train a decision tree classifier and then filter the predictions also using these features. However, ACR catalog does not provide computer codes for prediction, nor there are filters. AcrCrispr uses ACR distant homology with position scoring matrix and use support vector machines. So here we show the pros and cons of the 4 tools. AcrFinder is the only tool that can tell you not only if a protein is ACR or not, but also what subtype it is. However, it does not report a score or a rank. So ParCRISPR is super slow because it requires searching against UniProt to build a PSSM. So ACR catalog does not provide code for predictions or filtering. So last year we have combined AcrFinder, AcrRanker, and ParCRISPR to identify AcrACE operons in tens of thousands of prokaryotic and phage genomes. So the workflow starts with AcrFinder, and the resulting operon are further processed by AcrRanker and ParCRISPR, and also STS to remove false positives. So we have generated data sets with different confidence level. So about 1,500 operons are highly confident, the intersection, as they were supported by all the 3 programs and also found in self-targeting genomes. We have also shown that AcrACE operon are widely distributed in different phage and prokaryotic taxonomy groups. So all the pre-computed data are available on our website. So to summarize, I've shown you that anti-CRISPRs are natural inhibitors of CRISPR-Cas system in prokaryotic genomes and phage genomes. So ACRs have some unique sequence features that have been used to build a computational tools for ACR discovery, and 2 widely used approaches for ACR discovery, GLBIO association, and self-targeting spacer. I also shown you that AcrFinder combines GBA, STS, and homology into one pipeline, and AcrDB is a pre-computed AcrACE operon database that combines results from 3 different tools. I want to thank you for your attention, and I will be happy to take questions.